Thank you so much. I love the walk-up music. I feel hark back to my baseball days. So this is exciting for me. This is an opportunity to come back to AIS. Thank you so much, Grant, and the entire team at AIS at large. This is my opportunity to come back and share some additional progress at Acorn. It's been an exciting year for us, and let's dive right in. So I'm Drew Taylor. I'm the CEO of Acorn Biolabs, and we're talking about the cryopreservation of the hair follicle and its potential use in autologous regenerative medicine. Last year, when I was sharing our work, we were talking a little bit about the issue that I was facing back in my days at the University of Toronto and Mount Sinai Hospital, the Toronto version, where we were essentially trying to tissue engineer and regenerate connective tissues. That work essentially led to us identifying that the biggest limiting factor in our ability to leverage said tissues is the patient's age that they're coming from. And so if we're thinking about leveraging a patient's own cells in an autologous system, age is the biggest compounding factor in our ability to create these tissues on demand. And so that really led to the development of what I shared last year, a simple kit that can be used to essentially extract or pluck even hair follicles from the back of the scalp in practice in a very simple, non-invasive solution, where those cells can then be transported back to a centralized location. And I'm excited to say that after our presentation last year, we did make this available in the United States, and we've got a number of fantastic partners, some of them in this very room, where patients are able to actually lock in age on their side and have their own cells preserved. The other side, which I'll dive into a little bit more today, is how we plan on utilizing those cells. And it really is the other half of what we've created Acorn around, is not only the ability to capture a patient's cells, but then give them the opportunity to leverage those cells in your practices and deliver benefit. We think about this spectrum of how patients will use their cells on essentially a time scale of short, medium, and long term. And it's strictly related to how fast we think patients will start to leverage cells in these respective areas. And a lot of it has to do with the regulatory bodies and how long it takes to actually run trials to get to this level, and some of the science behind them and how fast it's progressing. In the long term, it is quite exciting, and we've got programs with a number of universities across Canada here, and now relationships into the US, where the cells that are harvested from the hair follicle, including fibroblasts, keratinocytes, and the MSCs present, can be turned into a number of different cell types. That reprogramming, obviously, is something that is manipulating those cells. And so from a regulatory perspective, that is a long cycle, and we're very frank, we think this future is probably 10 years away. But the cells from the hair follicle have been turned in our lab into neurons, pancreas cells, NK cells, immune cells, skin, cartilage, bone, and fat. On top of that, in the medium term, it's really looking at leveraging the actual cells that are present inside the hair follicle without that manipulation, so expansion, so minimally manipulated therapies. And this is actually great in the hair follicle because we have this multi-diverse population of cells where you've got applications and potential targets across dermatology, plastic surgery, wound healing, in aesthetic medicine, topically even, and then on top of that into orthopedics and sports med. What I'm going to present on today is really the short-term work that we have been doing on creating topical and then injectable solutions that don't leverage the cells to deliver back, but actually leverage the cells in the lab to be a factory or a resource that can continually produce a patient's own growth factors, exosomes, and matrix molecules on demand to be delivered back to that patient. Some of the work that we've done is across a number of different areas with the institutions. You'll see we're in Canada, so I want to give them a bit of a shout out. The NK work and neuronal work is going on at the National Research Council. We have a paper that is going out for pre-pub next month with the University of Toronto Mount Sinai Hospital on turning the hair follicle cells into pancreatic progenitor cells. And on top of that, with the University of Ottawa and Calgary, we're running animal studies in regeneration of cartilage using the bank cells from the hair follicle. In aesthetic medicine, we're very excited because we feel like this is eminent, and we're excited to share our first patient data, but the stage one of really this was to take that secretome that we were collecting as these cells are cultured in our laboratory and essentially characterize them. And we presented some of that early work last year where essentially we were able to create volumes of these growth factors and matrix molecules that far exceed what we find present in other autologous regenerative strategies like PRP. Step two was essentially to continue to do a phase one, essentially, where we did a RIP study, a repeat insult patch test. We executed on that this year and did this across 53 patients where we delivered this autologous secretomes AHFS, as we're calling it, an autologous hair follicle secretome onto patients topically. We got a scale of 0 out of 10 for redness and adverse events, and so we moved forward very quickly into testing this in post-procedure care. And so in step three, we had a number of patients. We have 8 to date that have done it across the back. We used 4 zones and used ablative laser to essentially test out our performance as a post-procedure care product against a simple HA, as well as a best-in-class product and a standard of care product. And then in step 4, which we'll talk about at the end, we're very excited to move this into the next stage beyond that with some members in the room today. And so our first clinical evaluation in this study that we did across a patient's back, we had a group essentially have 4 zones created across the back, applied this as a post-procedure topical, and tracked that over a period of up to 14 days. Really had an endpoint of 10 days where we saw the differences start to come together. But across those 8 patients, what we were able to essentially show was that not only was the AHFS able to reduce the look faster, including getting to a point where the patient was comfortable going outside because of scabbing and redness 4 days earlier than the comparatives, but really performed not only better than the standard of care that's used, but also best-in-class, multi-$100 solutions in post-procedure care. The really interesting part as well is because this is a biological product, is we ended up having a massive reduction in both pain, tenderness, and itch. So the patient was much more comfortable in the post-procedure care, which was quite exciting for us. To really zero in on their experience, we actually asked across the board, which of these zones was the one that you felt was the best on any given day, both from a pain perspective and from an evaluation perspective by the evaluator on redness and scabbing. And obviously, we had a 12x higher likelihood of being selected as that best zone. Unfortunately, we had this patient that was obviously having some big skin issues and needed a lot of help come in. I don't know if you can recognize him without the stubble. But in any case, we tested out our first series of patients on performance on the face after the study on the back. And what we were able to see is after 36 hours, all scabbing was reduced. There's no point spots of blood anymore on the AHFS side when compared to a standard of care. And then when we go to 4 days out, again, all scabbing was reduced. You can still see on the standard of care side that there is discoloration and scabbing along the temple and some of the marks from the laser itself. And so we're actually going to be expanding this number, and of course we're very excited to be working with a number of people, Dr. Franco, Dr. Cohen, who are here today, in starting to test this out in clinic in an early access period. And so I think the takeaway for today is really that if you are interested in evaluating an autologous product that is available in practice to leverage from your patient's own cells, that can be continually maintained essentially at that younger age as that patient continues to seek out opportunities in your practice throughout their lifetime. Think of this as really a way to lock in that age and have a consistent product similar to PRP in a growth factor spectrum and matrix molecule spectrum, but at much higher concentrated, elevated levels. We've got a full commitment to launch this in 2025, and so for the rest of this year, we're going to be continuing to ramp up those numbers and looking for physician partners to continue to help us with that goal across a number of different areas. And so I'd love to speak with anybody in the room that is interested in dialing in and learning more. Thank you so much.